Welcome to the deep dive.
Oh,
Okay, get ready because today we're zooming right in on this hidden battleground.
hmm
A microscopic struggle, really, between the tiny nematode Caenorhabditis elegans, the worm.
yep, C. elegans, classic lab organism.
Right, and these, well, remarkably clever predacious fungi.
It's a real David and Goliath situation, but on this incredibly tiny scale.
Exactly. And what we're digging into today is how this seemingly defenseless worm has evolved this really clever way to avoid, becoming lunch.
Precisely. We're essentially looking at a high-stakes drama playing out right there in the soil. Think of like a miniature action movie.
Huh, I like that.
And our mission, yours and ours, is to really understand these escape strategies. How does C. elegans actually get away from these fungal predators?
And what does that tell us about how behavior itself evolves? That's the bigger picture.
That's it, and prepare to be surprised, honestly. The survival skills of even these, apparently simple creatures are quite sophisticated.
Okay, let's start breaking this down. First up, the hunter, the predator in this microscopic arena.
Predacious fungi.
Yeah, and these aren't like the mushrooms you find in the supermarket. We're talking specific types.
Right, the ones that use these constricting hyphal rings, like
Yeah.
tiny microscopic lassos made of fungal cells.
Little lassos.
Yeah.
Okay. And the research we looked at really focuses on one species in particular.
Uh-huh. Drechslerella doedycoides. It's a key player in this story.
So how do these lasso traps actually work?
Well, this is what's fascinating. When a nematode, like our C. elegans, wriggles through one of these rings,
Yeah.
even just a gentle brush against the inside triggers this incredibly rapid inflation of the ring cells.
Whoa, like instantly.
Almost instantly.
Yeah.
Imagine a tiny airbag just deploying boom to snare the worm. There's even video of it. Movie S1 apparently shows it really clearly.
Okay, I need to see that. That speed.
Yeah,
It really highlights the challenge for the worm, doesn't it?
absolutely. The speed is remarkable. It's a split-second thing. It's almost unbelievable. And this isn't some new trick the fungus learned.
No, not at all. The sources point to evidence of this whole predator-prey relationship going back, wait for it, 100 million years.
100 million. Seriously.
Seriously, they've found these types of trapping fungi right alongside nematodes, preserved in ancient amber.
Wow. Okay, so this is a long evolutionary arms race we're talking about.
A very long one.
How do you think that long history might have, shaped both sides? The fungus getting better at trapping, the worm getting better at escaping?
Oh, undoubtedly. That's a crucial point. This deep history, this constant back and forth, it strongly suggests that interaction has been a major driver for adaptations on both sides.
Makes sense. Okay, so let's flip to the prey now. The worm itself, Caenorhabditis elegans.
Right, the intended meal.
For anyone less familiar, it's tiny, about 1 millimeter long.
Uh-huh. And it moves in that characteristic way, that sort of sinusoidal wave.
Yeah, that wiggle.
And it also has these exploratory head movements. It's kind of constantly sweeping its head side to side, almost like it's tasting or feeling its environment. You can see it in figure 1A.
Right, that distinctive wiggle and head sweep. But when it senses danger, like a gentle touch to its front end,
Ah, yes. It has a very specific escape response.
what does it do?
It reverses, backs up quickly, and crucially at the same time, it stops those exploratory head movements. Figure S1 details this.
Stops the head wiggles. Okay, so its feelers aren't just for finding food, they're part of this warning system too.
Exactly. And tuned specifically, it seems, to this kind of threat. It's a differential response, too, depending on where it's touched.
How so?
Well, touch the nose or the very front part of the body, it reverses. But if you touch it further back on the posterior end,
It goes forward.
it actually accelerates forward.
Hmm.
But the head movement suppression, that only seems to happen when it's backing away from a touch somewhere between its pharynx and its middle.
Interesting. So it's a very specific, coordinated reaction.
Extremely. And what's remarkable is that the whole neural circuit for this, the sensoromotor pathway, is actually really well understood in C. elegans.
Like we know the specific neurons involved.
Yeah, pretty much. Think of it like a simple reflex arc. Sensor detects the touch, signal goes to the worm's version of a brain.
Yeah.
Just a few key neurons, really,
Mm-hmm.
which then triggers the muscles for reversal and head suppression.
So a built-in danger, reverse smoothly program.
Kind of, yeah.
Okay, so the worm has this escape plan. How does it actually fare when it meets one of those fungal lassos? This is where size must matter, right?
Absolutely critical. Those fungal rings, their diameter is somewhere between, say, 10 and 25 micrometers. Tiny.
Okay, 10 to 25. And the worm?
Well, C. elegans starts life as a tiny L1 larva, maybe 12 micrometers wide, but it grows through stages up to an adult that's closer to maybe 50 micrometers across.
Ah, so there's a stage where the worm is just about the same size as the ring.
Exactly. And that's what figures 1D and 1E show. The L2 larval stage, the second stage, is the one most frequently caught.
Because they're the right size to just slip inside.
Precisely. Their body diameter often matches the ring size pretty well, making them likely to trigger that inflation as they move forward.
What about the bigger worms? Do they get caught?
Less often head first, they're usually too big. But interestingly, they can sometimes get snagged if they happen to reverse their tail end into a ring.
Ouch. But mostly it's the L2s.
Mostly L2s, and usually caught by the anterior part of their body, the front half. That seems to be the most vulnerable spot, according to figure 1E and that movie S1 again.
Okay, so let's picture it. Young L2 worm, perfect size for the noose, wriggles its head in. Sounds like game over.
You'd think so. But here's where it gets really interesting. The fungus, well, it doesn't snap shut instantaneously.
Ah, there's a delay.
Yes, a crucial delay. Figure 2A shows there's an average lag of about 5 seconds between the worm making contact and the trap actually inflating.
5 seconds. That doesn't sound like much.
To us, no. But for C. elegans, moving at its speed in that microscopic world, that's actually a pretty significant window of opportunity.
An eternity. Time to execute the escape plan.
Exactly. Time to hit reverse.
And do they? Do they actually get away often?
They do. It's
Yeah.
pretty amazing. The research shows that a whopping 81%, like 4 out of 5 wild-type L2 larvae that enter these rings manage to back out before the trap closes.
81%. That's a really high success rate against what sounds like a deadly trap.
It is impressive. And you can see this escape in action in movie S2. It just smoothly reverses out.
What do you think drove the evolution of such an effective strategy? The pressure must have been immense.
Oh, absolutely. That high success rate really highlights the strong selective pressure this predator must exert. If you can't escape, you don't pass on your genes. Simple as that.
And the key is sensing that touch, right?
That seemed to be the trigger. They tested this using mutant worms, one's called mech4 mutants. These guys have a specific problem with sensing gentle touch on their body.
Okay, so they're less sensitive.
Right. And when they put these mech4 mutants through the fungal gauntlet, their escape rate just plummeted, down to 43%.
Wow, nearly halved.
Yeah. It strongly suggests that feeling the ring starting to close, or maybe just the contact itself, is the critical cue to initiate that reversal. Get out now.
So it's the body touch, not necessarily bumping the ring with its nose.
Apparently not primarily the nose touch for this specific escape. They checked that too, using different mutants, OSM9 mutants, which have defects in certain sensory neurons in the nose. Their escape rate wasn't significantly affected in the same way.
Okay, so feeling the squeeze on the body triggers the backup command. But you mentioned something else was crucial, the head movements.
Yes, that suppression of the head wiggles during the reversal. It's not just a side effect. It seems to be a vital part of the strategy.
How do we know that?
Well, they looked at other mutants, worms that could still reverse perfectly fine,
Okay.
but had trouble stopping their head movements while backing up.
Ah, okay. Which mutants were these?
There were a couple. Tyramine deficient ones called TDC1. Tyramine is a neurotransmitter involved.
Sure.
And also LGC55 mutants, which have a defect in the receptor channel that responds to tyramine.
So these guys reverse, but their heads are still thrashing around.
Kind of, yeah. They don't suppress the head movements properly.
Yeah.
And even though they could back up just as fast as the normal worms,
Their escape rate was lower.
significantly lower. Only 49% for the TDC1 mutants and 60% for the LGC55 mutants managed to escape.
So just reversing isn't enough. You have to reverse smoothly.
Exactly. It highlights that the coordination is key. Suppressing
Yeah.
that head thrashing is essential for a clean getaway.
And they even checked worms that were bad at reversing altogether.
They did. UNC4 mutants, which have problems with the motor neurons needed for backward movement. Their escape rate was also lower, around 59%. It all points to needing that well-coordinated sequence. Sense, reverse quickly, and keep the head still.
Okay, so why? Why is keeping the head still so important? Why not just flail your way out? Seems counterintuitive.
It does a bit, doesn't it? But think about it. The researchers used a neat analogy, testing the worms with a nylon mesh.
Like a screen door material. Sort of, yeah, with small openings. Think of trying to pull a piece of thread through the eye of a very small needle. If the end of the thread is frayed and constantly wiggling around.
It's going to catch on the edges, make it harder to pull through.
Exactly. And figure 3 shows just that. Those LGC55 mutants, the ones that don't suppress their head movements,
Yeah.
they took significantly longer to get themselves out of a narrow opening in that nylon mesh compared to the normal wild type worms.
Ah, okay. That makes perfect sense now. All that extra wiggling just gives the fungal ring more chances to snag the worm as it's tightening.
Precisely. You're trying to escape this tightening news. Smooth, controlled withdrawal is what you need, not frantic thrashing that increases your profile.
Okay, that's a great visual. So did they test this idea directly with the fungus? Like, does head thrashing actually lead to getting caught more often?
They did. They ran competition experiments. They mixed the normal wild type L2 worms with the LGC 55 mutant worms, the head thrashers.
And put them all together with the fungus. Then they just counted who got caught.
And let me guess, the thrashers lost out.
You got it. Figures 4A and S4 show the result clearly. A significantly higher fraction of the LGC 55 mutants ended up trapped compared to the wild-type worms.
That's direct proof then. A clear selective disadvantage for worms that can't keep their heads still during escape. Natural selection in action right there.
Absolutely. Clear as day.
Did they do anything else to confirm it was really about the head suppression?
Yes, they did what are called rescue experiments. They took the LGC 55 mutants.
The ones with the faulty receptor.
Right. And they specifically restored the function of that LGC 55 gene, but only in the muscles responsible for suppressing the head movements.
Oh, clever. So they fixed just that one part of the problem.
Exactly. And what they found was a partial rescue of the escape defect. The worms got better at escaping again, not quite back to wild type levels, but significantly better than the untreated mutants.
That really nails it down then. It confirms it is the coordinated suppression of head movements linked to that specific gene and those specific muscles that's crucial for getting away.
It does. It ties the behavior, the genes, and the survival advantage together very neatly.
So just to recap this incredible microscopic drama, it's not just about feeling the fungus and hitting reverse.
No, it's much more elegant than that.
It's this incredibly well-timed, well-orchestrated sequence. Sense the touch on the body, trigger the immediate reversal,
Right.
and simultaneously quiet down those head movements to allow a smooth, efficient withdrawal.
All within that crucial 5-second window before the fungal trap slams shut.
Amazing. It really shows how natural selection can shape even what seems like a simple reflex into a highly sophisticated survival strategy.
It really does.
Yeah.
And the fact that you see this same kind of touch-induced head suppression in other related Caenorhabditis species.
Oh, it's not just C. elegans.
No, it seems conserved. That suggests it's an evolutionarily important trait, something valuable that's been kept around, likely honed over that huge time scale, that 100 million years we talked about.
Facing off against these fungi the whole time.
Constantly adapting.
And thinking about that long history, that evolutionary arms race. It really paints a picture, doesn't it? Each side evolving countermeasures.
Absolutely. The researchers even speculate that the constricting rings themselves might have evolved from earlier, maybe simpler, non-constricting traps, perhaps sticky nets or something.
So the fungus leveled up its trap and the worm leveled up its escape.
That's the idea of an arms race, yeah.
Yeah.
Constant pressure, constant innovation on both sides.
Which brings up a really interesting question for you listening in. If this level of complex, coordinated behavior evolved in response to a microscopic fungus,
Yeah, think about that.
how many other behaviors we see out there in nature, things that maybe look simple or random, are actually the result of these incredibly intense, long-running evolutionary pressures that we just don't usually see?
That's the real aha moment, isn't it? What looks like just a basic wiggle, simple escape reflex in this tiny worm,
Yeah.
turns out to be this incredibly fine-tuned survival strategy polished over millions of years, specifically to deal with this deadly fungal snare.
It really makes you wonder about all the hidden forces, the unseen evolutionary battles that are shaping life all around us, even down at the microbial level.
Indeed. You know, considering this ancient fight happening literally under our feet, in the soil,
Yeah.
it just prompts you to think, what other microscopic dramas are playing out?
Right. What other evolutionary test games are constantly shaping the creatures, big and small, that we share the planet with?
It's a humbling thought, really. Even in the simplest looking life forms, there are these intricate, fascinating stories of survival unfolding constantly.